Dear viewers, greetings. In this video, we are going to see about pH meter. This video contains the following contents. First, an introduction about pH, followed by pH meter, history of pH meter, principle of pH meter, types of pH meter, instrumentation of pH meter, steps for using a pH meter, applications of pH meter, advantages of pH meter, limitations of pH meter, and precautions for using a pH meter. pH. pH is a unit of measure that measures the acidity or alkalinity of a solution using a logarithmic scale with 7 as neutral, where low values are more acidic and higher ones are more alkaline. The pH scale ranges from 0 to 14, where the values below 7 represents acidity, values above 7 represent alkalinity, and pH of 7 is neutral. pH meter. A pH meter is an electronic device used to measure the acidity or alkalinity of a solution. A pH meter is a statistical tool that monitors the hydrogen ion activity in water-based solution, determining its acidity or alkalinity and represented as pH. The pH meter provides an accurate and quantitative value of pH levels of a substance, which is crucial for applications in chemistry, biology, environmental science, agriculture, food processing, and waste treatment. History of pH meter. In 1909, Danish chemist Søren Peder Lauritz Sørensen developed the concept of pH to describe the hydrogen ion concentration in a solution. The concept of glass electrode was first introduced in 1909 by Nobel Prize winning German chemist Fritz Haber together with Zygmunt Clemency Weiss. In 1934, the commercial pH meter was developed by Arnold Bachmann. Bachmann's invention stemmed from a need to measure the acidity of citrus products. This was a major breakthrough, leading to the wide application of pH measurements in scientific and industrial fields. From 1940s to 1950s, with advances in electronics, the design of the pH meter improved, becoming more accurate and easier to use. Principle of pH meter. The pH meter operates based on the electrochemical principle of a galvanic cell. A pH meter consists of a pH probe that transmits electric signals to the meter, which displays the pH of a solution. When the probe is submerged in the sample, hydrogen ions around the bulb replace metal ions from the sensor electrode, while some other ions transfer to the solution. The reference electrode maintains a constant voltage due to its low sensitivity to pH changes. The pH meter then converts the potential difference caused by the hydrogen ionic activity into a pH value by comparing the voltage with the reference electrode. Types of pH meter. There are 8 types of pH meter. They are benchtop pH meters, portable pH meters, pen type pH meters, continuous pH meters, micro pH meters, differential pH meters, ion sensitive field effect transistor pH meters or ISFET pH meters, and laboratory pH meters with multi-channel capability. The first type of pH meter is benchtop pH meters. Benchtop pH meters are stationary devices commonly used in laboratories. They offer high precision and are typically used for detailed scientific research or quality control. The second type of pH meter is portable pH meters. Portable pH meters are handheld devices designed for fieldwork and on-site measurements. They are compact, lightweight, and designed to be easily carried to various locations. The third type of pH meter is pen type pH meters. They are also called as pocket pH meters. The pen-type pH meters are compact and inexpensive pH meters designed for quick and easy pH measurements. They are highly portable and often used for routine testing. The fourth type of pH meters is continuous pH meters. The continuous pH meters are designed for continuous pH monitoring and control in industrial processes. They are typically installed in line with pipes or tanks to provide real-time data. The fifth type is micro pH meters. Micro pH meters are specialized devices designed for measuring pH in a very small volumes of samples, particularly in microliters. They are often used in biological and chemical research. The sixth type of pH meter is differential pH meters. Differential pH meters use multiple reference electrodes to measure pH and providing more stable and reliable measurements, especially in industrial and harsh environments. The seventh type of pH meter is ion sensitive field effect transistor pH meters. In short form, it is called as ISFET pH meters. The ISFET pH meters use semiconductor technology rather than glass electrodes, making them more robust and suitable for harsh environments. The eighth and final type of pH meter is laboratory pH meters with multi-channel capability. These are high-end benchtop pH meters capable of measuring pH simultaneously from multiple samples or locations. Instrumentation of pH meter. The instrumentation of pH meter contains three major parts. They are pH electrode, meter or display unit, and temperature sensor. The first part of the pH meter is pH electrode. Two types of electrodes are present in pH meter. They are glass electrode and reference electrode. Glass electrode is the primary sensing element of the pH meter, sensitive to hydrogen ion concentration. It consists of a thin glass membrane that interacts with hydrogen ions. The reference electrode usually contains a stable reference solution such as potassium chloride and is used to provide a constant potential against which the variable pH is measured. The second part of the pH meter is meter or display unit. The meter processes the signal from the electrodes, amplifies it, and converts it into a pH value which is displayed digitally. The third part is temperature sensor. Since pH is temperature dependent, modern pH meters may include a temperature sensor for automatic temperature compensation. In short form, it is called as ATC. Steps for using a pH meter. The first step is calibration. Calibrate the pH meter using a standard buffer solution, typically pH 4, 7 and 10. Rinse the electrode with distilled water between the measurements to avoid contamination. Ensure the meter displays the correct pH values for the buffer solution. The second step is sample preparation. Ensure the sample is homogeneous and well mixed. The sample should be at room temperature or the same temperature as the calibration buffers to minimize errors. The third step is measurement. Incubate, rinse the electrode with distilled water and gently blot it dry with a tissue. Immerse the electrode into the sample solution, ensuring it is fully submerged. Allow the pH meter to stabilize and record the pH value which is displayed on the meter. The fourth step is post measurement. After measurement, rinse the electrode with distilled water. Store the electrode in a suitable storage solution, usually pH 4 buffer or potassium chloride solution, to prevent it from drying out. Applications of pH meter. In environmental monitoring, the pH meter is used for measuring pH in water bodies, particularly lakes, rivers or oceans, to assess pollution levels or water quality. In agriculture, pH meter is used for checking soil pH to determine its suitability for different crops. Soil pH impacts nutrient availability and microbial activity. In biological and chemical research, pH meter is used for monitoring pH in various laboratory experiments, including enzyme reactions, cell cultures, and chemical synthesis, where pH plays a crucial role. In food industry, pH meter is used for measuring the pH during the food and beverage production. Example, dairy, brewing, and fermentation to ensure product quality and safety. In pharmaceuticals, pH meter is useful for maintaining the appropriate pH for drug formulations to ensure stability, bioavailability, and efficacy. In water treatment, pH meter is very helpful in monitoring and controlling the pH of water to ensure it is safe for consumption or industrial use. Advantages of pH meter. The advantages of pH meter includes precision and accuracy for high stakes applications, quick readings for real-time decision making, wide pH range for diverse testing needs, multifunctional for comprehensive solution analysis, portable and durable for field and industrial use, automatic temperature compensation for accurate readings, data logging for historical analysis and record keeping, reusable and cost effective, reducing the needs for disposables, and finally calibration options for long-term reliability. Limitations of pH meter. The limitations of pH meter includes calibration dependency for accuracy, electrode condition sensitivity and fragility, short electrode lifespan, temperature sensitivity affecting accuracy, interference from contaminants and non-aqueous solutions, drift over time causing fluctuations in readings, slow response time in certain solutions and situations, limited use in extreme pH ranges. Precautions for using pH meter. pH electrodes are sensitive and fragile, so one should not use them as a glass rod to stir the solution while measuring pH. Rinse and clean the electrode between uses. Store the electrode in proper storage solution. Avoid touching or damaging the glass bulb. Prevent over immersion and mechanical damage. Clean and replace the electrode as needed for reliable results. pH meters should be calibrated daily before use with the help of standard buffer solution. pH readings are temperature sensitive, so pH meters should not be exposed to sunlight. All the test tubes and glass apparatus used in measurement should be cleaned with distilled water before use. For each new sample, either uses a brand new fine dropper or glass rod or thoroughly wash the dropper or rod in water between uses. All the solution used in measurements should be freshly prepared. Dear viewers, that's all about the pH meter. Thank you for your support. Thank you.